Okay, welcome back, everybody. Can you hear me okay?
Yes.
Awesome. I hope that everybody had a wonderful time in the poster session. Our students worked really hard, amazing in the field, so very proud of them. Could somebody admit? Thank you so much. So if you didn't get a chance to meet Dr. Peace in the poster session, it's my privilege to be able to introduce him to you. So Dr. Peace is from Washington State University, and I know Cameron as a horticultural geneticist who knows all things tree fruit genetics. Cameron has his PhD in horticultural genetics from University of Queensland in Brisbane, Australia, and he's been at Washington State for about 15 years. Is that right? And he has a very active program in DNA-informed breeding. He's the co-director of the project Ross Breed, enabling marker-assisted breeding in Rosaceae. And many of his students and collaborators are joining us today via Zoom. And so I want to take a moment to welcome everybody who just joined us. Over the past year, I've been fortunate enough to learn from Cameron and the wider North American apple exploring community through the apple cultivar naming database and fruit registry meetings. The Fruit Registry is a really interesting app that helps folks all across North America to submit the locations and information of orchards from various fruit trees, and the app is actually currently being tested. The app that our students are developing is going to help feed that app as well, so we're very excited to be part of this group. And so Cameron today is going to share with us a really interesting genetic story about one tree in particular in El Dorado Canyon. You can learn more about this particular property in the homesteading family. It's the family of Jenny Pruden in the exhibit in the tree space. So I want everybody to please welcome Dr. Peace.
Thank you, Amy, and hope you can hear me all right. And Katie and Lisa for inviting me to be here today. So I'm just going to stand on this side. Okay, yes, I'm going to talk to you today about how genetically special is Grimes Golden Cross Wealthy. Something pretty specific. Okay, so two main sections of what we'll be talking about, or three if we include the Q&A. So first I'll just tell you about the DNA profiling system opportunity that we have and is available right now called My Apple Tree, we're calling it. Haven't bothered to register the name or anything, but whatever. But the main thing I'll be talking about is picking this particular tree that came from here and the genetic results, the genotypic results that have come out on this particular tree. And it's an example of anything. Even before the results arrived, I already was just going, I'll do it. Whatever the results are, whatever this tree is, I'm going to talk about this one. And so it is just a story that you can apply to anything, just following the same principles of any tree that's out there. So a side question I've got for you right now, I'm going to come back to this one in maybe about 10 minutes and ask you, this is for you guys, is how, or are Grimes Golden and Wealthy, which are in the title of this whole thing, are those even related to each other? So you can Google it now or whatever, so I'm going to come back and ask you guys, see if someone can check that out. Okay, so my main goals here are to demonstrate some of the capabilities of DNA profiling. Hopefully you'll get an idea of that from this, and demonstrate some genetics principles to you, particularly because I'm a geneticist at university and teach, and I'm passionate about making sure that people understand what genetics is, which is the process and consequences of inheritance. Okay, it's not the study of DNA, it's not the study of genes. Those things can come into it, great, but it's all about inheritance. Okay, that's the little pedigree diagram up there that captures what genetics is all about. Okay, so the first part then, DNA profiling with My Apple Tree. Okay, so we've got this thing that's, I've opened it up for the last couple of years or so to fruit enthusiasts in the country, where if they have a tree that's of interest, they get some leaf samples, send them to us, either in a tube or fresh tissue, and instructions are provided. And then that comes our way, we do some magic, and we figure out what it is, what the cultivar is, or better yet, its pedigree placement, its parents and things like that. So people are paying about $120 a sample to do this. And my research goals overall for this is to establish a single family tree of cultivated apples. So I think we can connect everything up into a single pedigree. I think pretty much all the cultivars out there are all related within about 10 generations. And then any tree that's done, any tree growing anywhere, some old heirloom one, old one on a homestead, or one in your backyard, or a seedling that you grow up, or just a cultivar from the supermarket, that can be determined to figure out where on the pedigree it is. Its placement in this big family tree. And while doing all this ancestry, it means we also are determining inherited attributes. We can do that too. And from that, determine, predict the performance of these individuals, of any individual. And so the work that I've been doing, particularly for breeding, has been a lot on the side of identifying the genetic factors underlying particular traits, especially fruit quality ones. And there'll be some of those I'll be talking about as we go through here. Okay, so yeah, we perform this magic. So the three main ingredients are the DNA profiling technology, historical germplasm that gets assessed, and the collaboration that's going on. And the flames are enthusiasm for all the people involved in this, and funding that we get. The more funding, the more we cook away there. But really, it's not magic, right? It's science that we're doing here. And so for the next bunch of slides, I'll just be trying to explain some of the mechanics of that, the science part of it. Okay, so we are looking at the DNA in this DNA profiling. And so DNA, as you know, is the blueprint of life, and it's got the coded instructions for how everything works in our bodies and any living organism. It differs slightly from individual to individual, is very important, and is the basis of genetics. It's inherited from parents, and it's arranged on chromosomes. So those are the three key principles that matter for understanding the next parts here. Okay, so let's look at one chromosome, of which Apple has 17. It's actually that picture of humans, but I just deleted a few and called it Apple. So let's say we're looking at chromosome 1. So the two pairs, the two members of each pair, every individual has inherited then one from the mother and one from the father. And they come together and give us our two. So we now have this DNA profiling technique called, it's based on SNPs. These markers called SNPs, genetic assays. This has been a key technological advance because it allows us to get the information of the genetic variation that exists in any individual from hundreds and hundreds of genetic points along each chromosome, and therefore thousands across the whole genome. And because we have such a high density of these, that provides some amazing power, lots of information there. So we have, to assay all these SNPs, we have these SNP arrays, or some people call them SNP chips. And so this allows us to test thousands of SNPs for any given individual and sample very efficiently. And also it's repeatable because we're testing the same ones over and over. Every time we run this, we're testing those same spots in the genome on other individuals. And so the particular SNPs that are on these arrays were chosen by fruit scientists, and 3 are available for Apple. The K there refers to the thousands of them that were put on. So we have 8,000, 20,000, 487,000. The green one in the middle, the 20K, that's the one that we most commonly use because it's just the most efficient, and we don't really need the high resolution for most of the things we use this for. So all the data I'll be talking about, the results I'll be talking about, are coming from data from the 20K SNP array. So these effectively are like a magnifying glass that allows us to go in and look at any part of a plant. Its leaf is typically what we use because the DNA is the same, right, throughout the organism, and look in and see what's going on in that particular individual from a genetic standpoint. So the main things I'll be talking about here are identity and parentage when I'll be talking about this particular individual. But it goes up further and further as you go up for the recent generations and how things are related behind that. You've got pedigree, and then way back behind that, you've got ancestry. But yeah, I won't be talking about those parts particularly today too much. So the SNP array data that we have, or that comes, is just binary. So it just comes in, yes, it's ACGT, but really it just can be converted to A or B. That's really all it is, just binary data. And so all on the chromosome then, we've got these hundreds, as I said, along every chromosome. We get the results back of the two letters for each individual. And so one letter will have come from one parent and one from the other parent. And so here's just an example of, like, right at the top of chromosome 1, we get, for running a sample, we get this data, and we know where in the genome it is. We know the order of all of these, where they've happened. So then we want to compare it to other things. So let's say we've done this test tree then, and we've got a whole bunch of things in the reference panel. And in this case, it's matching with something. It's identical to something in the reference panel. It's identical to Golden Delicious. Okay, so why do we know it's identical? Because it's matching, it's exactly the same letters at every one of these SNPs here, every one of these spots in the genome. Okay, so they're absolutely identical. Every time it's AA in one side, it's AA on the other. So completely identical entire genome. If something's not identical, it'll only be the same by chance in a few spots, but most of the rest of it will be different. So lots of differences there. And so we know it's not identical to those other things. If something is a parent or a child of our test sample, then it won't be identical, but it will share at least one of those letters at every spot throughout the whole genome. And that's, so we've got at least a partial match, right, for the entire genome. So every time, this one's AA here, so there must be an A over here. Yep, it's AB. Right? And this is because, for the parent-child thing, we're saying this is a parent-child relationship because whichever one is the child, it must have inherited one of those letters, which are alleles, variants of a spot in the genome, must have inherited one of those from each parent. So we apply that principle, and that allows us to determine which things are a parent or a child of a sample. If something is not a parent or a child, then again, there'll be some situations where just by chance it's the same for one of the letters, but in many other cases, it'll be different, many other positions. And so we know that it's not a parent or child. There are lots of mismatches. And if you look at each of those specifically, you'll see that the mismatches are where one is AA and the other one's BB. They're not sharing one of those alleles. And so one can't be the parent of the other, because every time something is an AA, it must pass on an A, or it must have got an A from both parents. So it can't have a BB direct pedigree match then. Okay, so parent or child, which is which came first, which one's the child out of what we're talking about here? So we can determine this if we know the age of the cultivars or trees of the two things we're comparing. Sometimes that is unclear. Having the other parent, though, in the reference data set really helps lock this in. So if we have the other parent, the orange there, and we look for all the situations of where one of the individuals out of the three we're talking about is AA, another one is BB, and the other one is AB. And so there are three ways around it that could possibly go, which are the three diagrams there, but only the one on the left makes sense. The other ones cannot happen. So that will lock it in. So in that case, we go, oh, well, good, great. We now know that the test individual on the left here is a child of the other one, the other two, in fact. So I use Excel particularly for this, even though there's software available, but I like all the colors and formulas and stuff you can do in Excel. So that's the way we do it. Colors, for example. And so here's our test sample is in blue there, but it's matching both letters, right, with Golden Delicious. And so it's got green and a 2 for each case there. Grimes Golden is a parent of Golden Delicious. And so it's matching at least one of the letters, so a yellow or a green. And then these other individuals that are children of Golden Delicious, so therefore children of our test sample, are also matching at least one of these letters. And so it's got no reds, right? And so only the individuals that aren't directly related, they're the ones that have some red patches here, all spread all through the genome, lots and lots of those. And so we know they're not a parent or child. So we have a reference data set that we use then to compare all the samples to. Any sample that comes in, we can compare with, and it's coming from lots of sources all over the world. That's that national, international collaboration that I was talking about before. And so it gives us a data set of more than 3,000 unique cultivars out there from more than 30 collections worldwide. So it makes it pretty powerful, but there are still new things coming up all the time, as I'll show in a bit. In fact, I'll show right now. So the aggregate results so far from the My Apple tree of about 600 samples coming on in. Well, some of them get submitted as a known cultivar on the left there. Yep. That left part, right? So 326 came in, someone said, you know, it's Grimes Golden, for example, or it's whatever they gave it a name. So half the time that became a new addition. We didn't have that one before. So Colorado Orange, for example. So then that would be one of these here. But other times it is, yeah, okay, fair enough. We already had that in the data set and it's matching. Other times for, you know, it's about a third here, it's incorrect. So the name that people think it is not right. Rootstocks pop up every now and then as well. And then also ones that people don't know, you know, what it is, or they know it's a seedling, or they have no idea what the cultivar could be. About half the time we can figure out what that cultivar is, and the other half of the time, again, many of them are seedlings, so we're not expecting them to be cultivars, but they are unique and they get added to the data set. So we now looking at the Boulder Apple Tree project, 8 samples were submitted earlier this year, and we unfortunately so far have only gotten results for 1, which is the one I'm going to be talking about. So I didn't have to choose then, which is the one to go with here. So thanks, Amy, for the photos of some of you guys out there playing with this particular tree, 498, and I'll just call it, what do you guys go, BATP? What do you call it, BATP? Calling it like that, just it makes it easier if I put where it's from at the beginning. So there we go, that's what I'll be calling this one from now on. Okay, so there we go, one of your photos. So the results so far, it's not matching any cultivar in the database. Okay, so that's of interest itself already, and its parents are Grimes Golden and Wealthy. That did come back. Okay, so are Grimes Golden and Wealthy at all related? Anyone? Anyone find anything on that? Too busy listening to me, wait. No one is Googling. Anyone have any idea or any feeling about that? Yep.
They are.
They are. Why? Why do you say that?
I guess.
Okay, so if you Google it, the parents unknown of Grimes Golden, and if you Google Wealthy, it says it comes from Siberian crab, right? From 1850. So, and Grimes Golden, what's that got to do with Siberian crab itself, right? So any Googling will say they're not related at all. So, okay, so here's these 2 parents, which I'll give 2 different colors coming in from here. So how genetically special is this particular individual? It's the second part here. Okay, so here we go. Grimes Golden cross Wealthy is leading to this individual. 498, make it easier. Okay, so imagine we're looking at all 17 chromosomes here, but I've just zoomed in on one of them, and I'll continue to repeat this particular image here. You know, it's a combination of the parents, right? So it has to have inherited one of its members of each of the pairs of its chromosomes there from each of those 2 parents. But you'll see that on each side, it's a mix, right, of the 2 grandparental parts. There's those 2 different shades that you see of the orangey, the gold, and the purple over on the right there. Okay, all right. So how special is this? An individual like this? Well, it's very special, of course, but it's unique. Okay, that might be an answer that you give. Okay, so let's say we had hypothetically 5 other full siblings that had the same 2 parents. They would be a different genetic mosaic, right? For that same chromosome pair, they would be different. In fact, the chance of getting something that comes up that looks almost exactly the same, something like 1% difference, within 1% different all around, is 1, I calculated this, 1 in 20 million trillion. So it's 2 to the 1 in 2 to the power 43. So yeah, that's very, very unique. Okay, fair enough. Another reason something could be special, you could say this individual is special, is because it's cultivar, whatever happens to be, right? So the principle behind that is there's a lot of historical value in the human experience with a particular cultivar. So there was the creation and the discovery and the raising of the original tree and all the stories and everything about that. There's all the perpetuation and distribution of the grafted clones that has gone on, and all the industry and all various things that have gone on regarding that. Lots of stories involved there. And anyone who's ever grown the tree, enjoyed its fruit, and been influenced anyway by its existence, that all means that that's really makes that individual very important. No matter what the copy of it that you have. You know, you have this clone, but still that ties you in with that cultivar throughout everywhere it's been grown. So yeah, that's got huge significance, I agree. And it's the motivation, the inspiration for many of us, these particular cultivars and their names. And it sure is for me. However, this is historical specialness, if I just want to draw the line here, rather than genetic, right? So I'm talking about the genetic here. So yeah, very much so, I acknowledge this, but that's not the genetics. And it turns out anyway, this 498 doesn't match, right, the identity with any cultivar in our reference database, as I said earlier, but that does give the opportunity for some historical sleuthing to find out what it is, because it does match. It is identical apparently with something from elsewhere, another tree in Colorado. So that already shows that it was a grafted tree, and it probably was a cultivar. What was its name? Right, so someone find out. This is a really great one to go after. So another reason it would be special is because it's a child of cultivar Y. Okay, so in this case, cultivar Y has its own historical significance, right? So any known children automatically tie in with that history, and likely it's inherited. We hope it's inherited some useful attributes from whatever that parent is as well. So these are genetic because this is the child of, right? It's inheritance. So it's tying in with its parents. And so, yeah, so you get the history being inherited in this individual here. So, okay, fair enough. It depends, of course, on the historical value of that parent. And it does depend on us knowing that there is that parent-child relationship. And it depends on what was actually inherited from that particular parent as well. That helps give some specialness to it. So if we take that back further behind cultivar Y parent, we're going into the ancestors on one side of the family there, then it's special because it's descendant of all those other ancestors. So it's tied as well, just like it was to the parent, it's tied to the histories of all those other cultivars as well, all those ancestors. And of course, it might have inherited some useful attributes from each of those ancestors also. So just as it was for the parents, it depends on the historical value of each of those known ancestors, and it depends on what was inherited from each of those. And similarly, again, what about close relatives, things that have come off those ancestors? So yeah, it's also special because it's related to these other things, not directly descended from, but close relative of a bunch of things. So it might have inherited some useful attributes that also these relatives inherited. So that would give it some specialness, some genetic specialness. And it's somewhat tied to the histories of those close relatives, right? So if you have an uncle that's really famous for whatever, you know, you got a little bit of a foot in the door of being part of that specialness as well. Or your sisters are, you know, really cool and you like to tell stories about her. So it depends on the historical value of those known lateral relatives. And it depends on what was actually inherited from the ancestors and shared that your individual 498 shares with those lateral relatives. Okay, so let's apply this now to our individual, which is from Grimes, Golden, and Wealthy. So yes, 498 is a child of Grimes, Golden. So what's Grimes, Golden? Well, that arose in the late 1700s. It's been around a long time. It has lots of interesting things. It looks beautiful. It's yellowish flesh, etc. Tastes really interesting. It's quite nice. Got lots of great attributes about its fruit quality. It's got some disease tolerances and a whole bunch of susceptibilities. Does it have a historic role in Boulder? Yeah. So I imagine it's got something for, first of all, for the kid to be around here. So that's got some interesting interest to it. It's also Grimes Golden. Its claim to fame in the apple geneticist world is that it's a parent of Golden Delicious. We don't know any other children actually of Grimes Golden, but parent of Golden Delicious, as I say, Golden Delicious is one of the highest production cultivars in the world, right? So it's rural economic prosperity for lots of people. It's delivered a lot of health to a lot of people all around the world. So that's amazing. So 498 is half sibling of that. So it can boast about that. And Golden Delicious is also a very common recent ancestor of most modern cultivars as well. And there are some desirable alleles that Grimes Golden has given to Goldens and Delicious. And so I've got a few of them here, which I'll explain in a bit, but this is about aroma and storage and acidity. So 498 and Golden Delicious inherited the same ones from Golden Delicious that I'm showing here, including the low blush or no blush stripes version here. 498 also got some other things from Golden Delicious, including a bunch of ones I put in red there, which are undesirable because they lead to suboptimal storability and firmness. What about the ancestry of Grimes Golden? Well, mostly we don't know the parents, or we don't know the parents at all, actually, of Grimes Golden, but what we have been able to determine is one of the grandparents is Rainette Frunch. Okay, so this is going to come up a bit, this particular individual. So this is the most common known ancestor of heritage and modern cultivars. It's in the background of everything, pretty much. It's a likely source of many of the desirable alleles, gene variants that in cultivars that we enjoy. It arose more than 500 years ago in Normandy. And it's the direct parent, first of all, of many very old cultivars, and then the ancestor of, as I said, just about everything. So we can track through the genome, and I was able to figure out that 498 inherited some particular things via Grimes Golden from Rainette Frunch, including a low acidity allele, a high acidity allele, the no blushness, the allele for that, it inherited that, and then the aroma, the good aroma, apple aroma, it inherited that too. And let's look at the other parent, Wealthy. So 498 is a kid of Wealthy. Well, Wealthy was raised in the mid-1850s in Minnesota by Peter Gideon. As I was saying before, it was said to be from Siberian crab. It's very winter hardy, as its biggest claim to fame. So it survived when so many other things didn't. And it had a historic role involved as well. So that's another interesting thing to check out. And there are some desirable alleles that 498 got in fruit quality, especially for firmness and storability, from Wealthy. Got some other things from Wealthy as well, including some less desirable. Now, Wealthy is not from Siberian crab. We've been able to determine its ancestry, and its two parents are Duchess and Jonathan, Duchess of Oldenburg and Jonathan. And Jonathan, we know even behind that one as well. We know Jonathan is a seedling of Circus Spitzenberg, which has been around even longer. And Circus Spitzenberg is a child of Rainette Frunch. So Rainette Frunch is coming on in again to 498. And again, we can track through what it's inherited from each of these ancestors. And another way you can say that this individual is special is because it's those two parents combined. That particular combination is really cool. So the principle behind this is that a child is the best of its two parents, right? Which each of us are, right? We're the very best of our parents. And it's the major principle underlying, major thing behind breeding, right? Is breeders think I should cross this with this because they're trying to get that combination of those two things together. It's not really true though that it's the best, right? You have exceptional luck if an individual has inherited the best of its two parents. It could be achieved with selection among siblings. If you have lots and lots of siblings, hundreds, thousands, etc., then which is the other main aspect of breeding, then you can pick out the ones that have, or the one that has inherited all the best stuff. Is that what happened to 498? Well, the more stringent you are with selection, the more likely you can find those superior children that are the best of everything. If 498's a cultivar, which probably is, okay, then it did go through some selection to be bothered, for the people to bother to graft it up and spread it around and be eating this, etc. So some selection. So we'd expect it to be a bit better than a random child. So it does depend on what was actually inherited as well. And so here's that, adding everything together from what I was showing you before about the fruit quality and other things, this is what 498 has. So it ends up getting for example, in the second line here, it has a blush allele and that low blush allele that I got from Rainette Frunch. When you put those together, you get some blush or stripes. And so that's what it actually is. Here's the photo of it. And it's typical acidity I'd expect from it. It's probably not a winter apple because it doesn't have the best storability alleles. It's probably not very firm, doesn't have very good there. But I expect it has a nice apple aroma, because it's got one of the alleles for that, and that's dominant over the one that doesn't. So it should be aromatic. If it's not so unique, then it's not so special, right? So when parents are related, then there's more sameness. And we already saw that Raynet Frunch is in the background of both of these, right? So it's not, we don't have separate colors therefore coming through here, which would mean we'd get some similarities. And so it's, once the parents are related, it's possible to get that combination in multiple children, or similar combinations would come up more often. And some ancestors are common among cultivars. So Golden Malicious is, as I was saying, is a common ancestor of lots of cultivars, and Raynet Frunch is pretty much a common ancestor of just about any cultivar out there. So that's going to make a lot of similarities among cultivars. So we can look at this, and Raynet Frunch is a grandparent of Grimes Golden. So we expect Grimes Golden to have about a quarter Raynet Frunch in it, and it's a great grandparent of Wealthy. So we'd expect an eighth. If any of you are genealogists, then you'll be able to quickly see that that means they're half first cousins once removed. And so we'd expect there to be about 3% of Raynet Frunch shared between Grimes Golden and Wealthy. Okay, that's what we should get. And that's from what we've been able to determine from this ancestry of Raynet, of these two cultivars from all this DNA profiling work. So it turns out, actually, they share way more than 3%. They share 33%. Okay, so 10, 11 times as much as we'd expect. And that's even with the discoveries we've made about the pedigree here. So how's that possible, right? Well, Raynet Frunch is coming into the ancestry of Wealthy and Grimes Golden, not just through what I was showing you before. So with the other parent of Grimes Golden, you know, it's probably 3/4 Raynet Frunch itself. And then Wealthy, there were some other ancestors I didn't mention in there. It's coming in there to a lot. So this is making, even just through the Raynet Frunch ancestry, it's saying that these two cultivars are actually closer than half-6. And if you add on any other common ancestors that they do have, they're probably the equivalent of full siblings, which is crazy. So yeah, cultivars are, there's a huge amount of recent shared ancestry that is not yet represented by our currently known pedigrees. And this is not special. It's not something anything particular close between these two cultivars, Grimes Golden and Wealthy. This is very random and very normal among cultivars. So in fact, modern cultivars, there's a lot of relatedness among those, as you probably hear, etc. all the time. And it's the equivalent of everything being on like one old town village street, right? So everyone's a close relation. But when we go out to heirloom cultivars, that's particularly because of Golden Malicious. When we go to heirloom cultivars, they're still really close. There's still so many close family ties, as I was just showing you there with Grimes Golden and Wealthy. And so it's like you're in a small village there, not really adding on that much more unusualness. So there's already a lot of unusualness just among modern cultivars, is another way to say it. And that's especially thanks to Raynet Frunch and a couple of other ancestors, but particularly that one being in the background of so many things. So yes, indeed, there's a lot of relatedness among apple cultivars. And so therefore, each individual is nowhere as unique as that 20 million trillion, especially if the parents are well connected. Some of that sameness, though, is probably needed, right, for human use. So if you don't have that, maybe you don't even get fruit on the tree. So it doesn't mean it's a bad thing. So just because it's clearly inbred, it doesn't mean that that's always bad. So, okay, maybe it's special because it has some differences then. Okay, so despite having these ancestors in common with most other cultivars, surely it's got something unique, right, that it's inherited, something it's got that other things don't really have. Because, of course, each child inherits only a random half of each of its parents' alleles. And so, related doesn't mean identical. So it still should have some of its own stuff to it. But the more children that a cultivar has, the more likely that its alleles have been maintained in later generations. And all those ancestors that I was talking about before of 498 do have lots and lots of children and descendants. So yeah, probably you wouldn't expect there to be too much unusual, too much unique within 498, as it turns out. So let's have a look at that. Just doing this different view here of the 17 chromosomes now arranged in a circular way. Everything that this individual 498 has inherited from Grimes Golden is in the inner circle, and from Wealthy is in the outer circle. So Grimes Golden, we know a bit of its ancestry, right? The Raynet Frunch as one of the grandparents. And so those are the blue parts. Now, we don't expect a quarter in there, but you can see there's way more than a quarter inside that. And so that is indicating that some of these other ancestors here probably have must have a lot of rainet frunch in them. And then wealthy is all these other things. And here's the mosaic of what wealthy has contributed to 498. This is a bit more what we'd expect. So the rainet frunch is about an eighth in there, and the other parent of Ahsoka Spitzenberg is about an eighth, and the other parent of Jonathan, the light green, is about a quarter, etc. And then a half of that outer circle is Duchess of Oldenburg, the purple. These are three regions that are identical. It's the same bit of rainet frunch has been inherited from both sides. So it's homozygous in those regions, 498 is. We can look at the trait locus alleles that this individual has inherited. So I've spotted those on here, and just focusing on a particular, some like the S locus for cross compatibility and that fruity aroma, and then the skin color. And then if you look at the color that's underneath any of those, the ancestral segment there, you can figure out where it's come from therefore. In the rest of the genome somewhere, there can be genetic factors for all sorts of other things. So such as tree hardiness, right? This tree has been around a long, long time. It's been withstanding many decades of stresses. So where is that, right? To be determined. Don't know yet. So let's compare it rather than with everything, because it won't have any differences with those two parents, but let's compare it with the top 10 production cultivars in the US. And these top 10 cover about just over 80% of all the production in the country. And so spotted on in stars here are all the regions, all the spots where there is an allele, something that 498 has, that none of those other cultivars have, the top 10 have. Okay, so that's good, because that's additions to genetic diversity if 498 was suddenly to become the 11th, modern cultivar or cultivar grown at the moment. So, okay, it's got a little bit of stuff that other things don't have, which could have various uses, various influences and value that is not in the top 10. And maybe it's great because of its potential as a parent. So maybe it's accumulated lots and lots of favorable alleles, which is great. Maybe it will combine really well with a certain other parent. Maybe it has rare sets of linked alleles. And so let's look at that in here. If we zoom into this spot here, it could be, for example, that rare one that it's got that none of the top 10 have, could be for disease resistance. And it has that. And it also has this common one that everything else has. Let's say it's for annual bearing. And so all the other cultivars have got the annual bearing, and they've got the susceptibility allele that was next to it, which came along for the ride. And there was this recombination here, this change in it that allowed, now we have some resistance, right, to come on through. That's always going to be connected now with this. So that would be valuable if that occurred. And so it's worth zooming into each of those unique spots to see what's going on with this individual. Okay, so this is just in summary then. So how genetically special is it? Well, here's all the reasons why it would be, and some of the thoughts about that, right? So it's a unique genetic mosaic, yep, but it's closely related to lots of other cultivars. It's a child of the historic Grimes Golden, great. It's a child of wealthy, yep, that's really good. It's a child of Grimes Golden and wealthy combined. There are no other cultivars out there known in those 3,000 plus, right, that's a child of those two. It's inherited some favorable alleles, although it's not an ideal accumulation of them. And it's inherited some rare alleles that are not in the current top 10, and some possibly useful linked sets of material there. So that could be useful as well. Of course, it's historically valuable in itself and in the context of other historic cultivars in this region. So I'd like to acknowledge folks that put on as co-authors here. So Sten is a postdoc in my lab. Nick Howard is a researcher was at University of Minnesota. Now he's in the Netherlands, who is accumulating all the data sets here. And AJ and Ty are researchers and students in my lab. And of course, thanks to you guys for being involved, not only for sending this sampling to make this fun and inviting me here, but also all the collaborations that we have had till now and will continue to have. So with that, not too much time left, but questions, but Q&A. Okay, we have a question over here. Someone got in early. Yes.
Yeah, you mentioned you wrote that slide that showed that, hey, if apples were, related, they'd all be from the same street. You look at like, what's generally available in the top 10. And then if you out from that with heirlooms, you look at like same small village, right?
Right.
So I'm curious if those trends that you see, does that relate, per country? Like you went to Asia and you look at the lineage of apples in Asia, or other places in the world, you see different trends, or like this area has much greater or much less diversity in genetics.
Yeah, right. Nice question. So yeah, is this just because of the particular history of the US, of how everything's come through and been inbred? So the answer is, I'm talking about the world. Yeah. So the only reason somewhere might be a bit different would be if for some reason, and they haven't, but have incorporated a whole bunch of wild species or something like that. And there are a lot of wild species in China, for example, and they have a huge apple industry and they do breeding, but it's based on same cultivars, Fuji, Golden Delicious, etc. It's the same old stuff, Honeycrisp.
So that's a worldwide species.
So yes, absolutely what I'm saying is worldwide.
When did apple drafting method begin?
Roman times, apparently.
Oh,
Yeah.
so I was going to ask you, if you visit an old tree, is it guaranteed to be drafted? Does anybody ever look at the DNA of the son versus the rootstock? And I mean, because you can't tell with an old tree.
Right. Yes. So, okay, it's a couple of questions in there, but yeah, the rootstock versus when you can't tell. When you can tell, you see a graft line, and we're pretty sure it's a graft line, or that's one way to tell. Another way is multiple samples are showing the same thing. Multiple trees, I should say, are identical with each other. Okay, there must be clonal propagation at that point, such as 498 with another tree elsewhere in the state. But also, if we test the rootstock, have a shoot coming up or something like that, or we're trying to get into the roots now, for example, and test and compare the DNA, then that's another way of checking. So if it was the same, then it'd be like, oh, no, it's a seedling tree. But it's the rootstock itself that's now the tree.
The reason I wanted to ask that is because I would suspect a lot of the older trees are growing from the seeds. The Johnny apple seed there. No?
There are not too many left of those anymore. So when you say the old trees, pretty much talking about grafted cultivars that were in orchards. There's just about nothing left of the original Johnny Appleseed seed planted stuff. Yeah. Although cultivars like Grange Golden and Wealthy and things like this, are themselves, those sorts of cultivars are themselves Johnny Appleseed things in many cases. But they were originally just part of a seedling orchard, and then they were selected because they were great. So it's not like we've lost all that. We do have a lot of the best of that in the existing cultivars, hundreds of thousands.
So when you study the ancestry, it's all about the science.
Oh, we're also doing rootstocks as well. That's also quite interesting and very revealing. And so, yeah, there are particular, often it's the same sorts of cultivars or certain dessert cultivars that then got used, like Cactus, for example, Winter Hardy, which is a parent of Wealthy, but also its seedlings as rootstocks also carry Winter Hardiness, for example. So that was a common one. And certain other dessert cultivars were used as rootstocks as well. And so then there'll be children of it, not the clone rootstock, but the Duchess cross something will be the rootstocks. Yeah, so we're also looking at rootstocks. Yeah, it's fascinating. And just the rootstocks itself and doing analysis of all that can be really interesting in understanding the history of what's going on and where that material came from.
When you perform this genetic testing, how often do you come across unknown cultivars? Since you showed data at the very beginning that was just our data in particular, correct? So how often does this happen?
So yeah, so if you recall those two pie charts I put up, there was when when something's named something. So so let's say this particular 498 actually was, you knew the name, but the name wasn't anywhere else, and whatever it was called, right? Boulder Terrific or something that wasn't quite from right here, but that would have come up as, about half of the things that get submitted are like that. It's like, cool, okay, now we have, Boulder Terrific. And it's now there. Hopefully there's a watercolor for it as well, but now we have it. There's a bit more rigor in making sure that really is that watercolor one, for example. In other cases, it might be the first time that name's ever come up, or someone's just named it themselves. And that's
about half of what you just.
at the moment, yeah. And it's going to obviously get less and less as we, get more and more, especially through my Apple Tree. It's why I'm doing this actually with my Apple Tree, is to get a hold of all this US material that's out there, because there's still a couple thousand cultivars out there still to be added. Yeah.
Do you use genome-wide association studies to work frequently? If so, why? And that kind of the second part.
Yeah. So I don't personally, but colleagues do. They use the SNP array information to then associate that, and they collect a whole lot of performance information, a lot of material, and they do this array analysis, get the data, the genotyping on a lot of material, and then use that to do what you said, a genome-wide association analysis, which is to identify where in the genome are the positions that are influencing the traits, to help identify those. So yeah, there's a lot of that going on. In fact, the 487K array was designed for that much higher resolution. So if someone wanted to do that sort of study, that would be actually the array to use.
Could you then share each genome-wide association study for each cultivar and find potential lineage in relation to that?
So what you're talking about is just an association of trait and phenotype with a bunch of material. So all you need to do is do the lower resolution, the 20K, for example, which is still really high resolution, but do that one to do what you're saying, to figure out what's got what, like the alleles are showing, how everything's related to everything else. Yeah, so the genome-wide association studies only need to be done, once by a research program somewhere, and then that information gets usable. So a lot of those jewels I was putting on, like some of these things here, came from those sorts of studies. Yeah, and we accumulate all of that to add more and more and more to this. Only put the fruit quality ones, a few of the fruit quality ones.
So I think that the discussion about the recombination and the combination of the unique alleles is just super exciting and cool. And I was curious, in terms of the implications of this work, what do you see happening from this work? Like let's say she found a bunch of really wonderfully unique recombination sites. Like, would this be more useful to sort of local growers and small scale and heirloom markets, or to broader agriculture, or both?
Yeah, both.
Yeah.
Yeah,
Can you explain how you see that?
The answer's both. So with all this information, as we're collecting it, getting it all together, and if we get some good programs, etc., then there'll be a database available to anyone who wants to exploit these existing cultivars, to know everything about them, or these existing trees, if it's a new seedling tree, or use them as parents. And when you want to use something as a parent, you want to combine it with something. So I was talking about the complementarity before, for example. So it might be 498 ends up being someone in Washington who wants to do a cross with something and get to a particular target. They design, the idea what they want it to look like in the end, and then query the whole database and finds out this one would be, if you match it with that, you'll have a very high proportion of seedlings will get what you want. And so they say, oh, who's got that tree? Can I get a hold of that?
So if I look at your database and hypothetically say, okay, I want a fire blight resistant and crisp tart apple, and potentially figure out how to read that.
With as much diversity in its background as possible. For those particular things, and also for the rest of the genome where we don't know what's going on, just mix it up, have it all different, and I don't want any ducks in the background, and those sorts of things could be what people do. So we're moving towards that, and going after funding that will have the programming to be able to do those sorts of intense analyses.
Maybe I might probably
transition if there's any last minute questions here.
I have just one. I'd probably throw it at you, so. Just, what else is there cool about 498?
498 is pretty fantastic. It's down Eldorado Creek, and it's part of, I just lost the word, not heritage.
Homestead?
Homestead, thank you, from the Jenny Drew family.
Okay.
And so it's really fantastic, and it has the most amazing view of Flatirons. This tree has one of just the best views ever. It's really fantastic. The property owners told us it still makes really great fruit. When our fellow researcher and I visited in the wintertime, it still had abundant birds visiting the tree. So it's a really great wildlife tree. It has great views. It has great tasting apples. And it has wonderful history in Boulder. So I love this tree. And it's grafted very well. We've been able to get approximately 10 grafts off of this tree and back out. So hopefully we'll get more. The other really cool thing is it matches a tree down in Cortez that Montezuma Orchard Restoration Project is also found. And it also matches a tree in Montana. So we know
Those are the kind of facts I was curious about.
Yes.
That's great.
it has friends.
It's truly a lost cultivar that has been rediscovered potentially.
What is its name? What was its name? And is its name, right? Find out, someone, please.
Thank
Yeah.
you. All right, so I would like to keep us rolling here to our last event for the evening. So I want to introduce two very special people. So we're going to have two panelists tonight who are going to talk about things at the nexus of human, wildlife, and apple interactions. And I'd first like to introduce Megan Bose. So Megan is a restoration plant ecologist with Boulder Open Space and Mountain Parks, and she's worked across multiple different types of landscapes, including urban, forested, and historic, and is a longtime supporter of the Boulder Apple Tree Project. So Megan is the reason that we can do our research, and she has been instrumental in getting us the permitting and helping us to access the different lands on which we actually do our research. Megan is also a really wonderful contributor to the oral histories of the project and has collected those and helped us. We have now 2 oral histories cataloged with the Carnegie Library, and so three oral histories.
And I think there are others as well.
Oh, yeah. So absolutely fantastic contributor to our project and an expert in thinking about some of the interactions that we have here. And so if you could take a seat, our panelists are going to sit over here. And then I'd also like to introduce Blair Norman from, who's representing 2 different community groups tonight. So she is the president of Community Fruit Rescue, which is a local nonprofit that harvests and rescues fruit for local food shares, animal sanctuaries, and various fundraisers and other nonprofits. And they've rescued over 10,000 pounds. You want to be 26,000. Oh, excuse me. They've rescued 26,000 pounds of fruit this year, which is incredible. And Blair's also representing the Boulder Bear Coalition today, which strives to reduce the human-bear conflicts and to keep local black bears free and wild and keep them out of the community with various techniques, including the reduction of attractants. So we have these fabulous panelists here today, and I'd like you both to maybe just get started by telling us a little bit about your positions and what you do with respect to the interactions between humans, wildlife, and the fruit trees. So Blair, do you want to go ahead and start us off, and then we'll go ahead and hear from Megan on that, and then we'll just take questions after that.
Sure. So I, along with 4 other Boulder groups, started Community Fruit Rescue in 2014 when we saw an abundance. It wasn't quite this big of a fruit year, but we saw apples on all these trees, and different groups came together saying, what can we do about this? It's just falling to the ground. My kind of more concern was how can we make this fruit more available in February than just right now. Other Brenda, who is started Boulder Bear Coalition, came together, and there were lots of bears sitting in the trees. There was human bear conflict, and the bears were breaking the limbs of the trees and just not wanting to leave. And then Boulder Food Rescue came in and said, how can we get this food out to our community? So, yeah, so we just started getting volunteers, going to homeowners' houses, picking apple trees, and then sharing the fruit with the different homeless shelters, food pantries in the Boulder area. So, and then Boulder Bear Coalition started in 2012, Brenda, there was a bear had been put down in her neighborhood, and she was like, "Why?" And no one could really answer the question. And she's like, "Well, why are bears even here in Boulder?" And she hadn't really noticed the fruit, and the bears were coming in for the fruit and for the trash. And so she started bear sitting with CPW, Colorado Parks and Wildlife, and to learn more about Colorado Parks and Wildlife position in their concept and thought, and what their goal was with keeping bears, you know, the human conflict in Boulder. And so she got that information and was like, you know what, there's no one really trying to advocate. The city was letting CPW just handle crisis management. And there was no one in town trying to educate the humans as to why the bears were having to be relocated. And so she started Boulder Bear Coalition to get everybody kind of more on board with we're here in their in our backyards.
Thanks for having me tonight. My name is Becky Bowes. I'm a plant ecologist with the City Boulder's Open Space and Mountain Parks Department. I have been responsible for managing our ecological restoration program since 2009. The department has obviously been doing restoration going back to the 90s, if not before, but we really wanted to ramp up efforts and hire additional staff to perform those. In addition to permitting research on open space and mountain parks, I've participated in a couple of blitzes. I've also partnered with the Boulder Bear Coalition. We've done, I think, 3 plantings over the last 4 or 5 years to try and plant berry producing shrubs that will encourage the bears to stay in town or in the woods and not in town. We also, there's kind of an unspoken rule, if you will, that we will not remove non-native apple trees in the forest. We'll keep that as foraging bears and other wildlife species, but we probably are going to be slowly removing them on the plains, again, to keep them out of town. I can talk, there were a handful of questions that came through about specific wildlife concerns or issues that folks might have, and so I can talk more about that, but it's been a pleasure to both work with the Apple Tree Project and the Boulder Bear Coalition.
So what we'll do is we'll just go ahead and open it up for questions. And I know that many students have brainstormed some questions for our panelists. So if people just want to go ahead and start asking questions, we'll open it up first mainly to questions sort of about the human wildlife fruit interactions, and then also questions about the way that these 2 wonderful people got into this work, which I think would be really informative for a lot of our students here tonight. We're hoping to get careers and to be involved in things like you. So go for it. Any questions? Okay, we'll go ahead and
So you said that you've been part of like the restoration program since 2009. Were you part of anything else before that? Or how did you career?
Yeah, I've been with the department since 2004. The other hat I wear is vegetation community mapping. So I'm responsible for managing that with our other plant ecologists on staff, as well as we're plant surveys and things of that nature. But we really tried to ramp up the restoration efforts starting in 2009. And we've got a very successful program. One of the things that we've been very successful at is working with Boulder County Parks and Open Space and other municipalities with collecting native grass and wildflower seed, having a grow out increase. So we can only collect 5 pounds, but we can potentially grow out 500 pounds. We've got one collection coming in possibly today from a grower in Washington State, and he was able to grow 1,500 pounds of wheatgrass for us, which is a pretty big increase.
So the restoration signs we see when we're hiking the open space is your department?
Yes.
I stay off this area too.
Yes.
Right, so Tammy. This actually just because I have the answer to that question, but I'm asking, are you doing like GIS work as well?
Yes, so we actually have tablets that we take in the field and we're digitizing in the field. We used to do it on paper and then we had to scan things in and, you know, rest of the conversion, and it was a big process. So now it's much more readily available, and it can then theoretically be, once it gets uploaded to the system, it can be ready to use on the fly.
That is super cool. I'm very interested in GIS, so I was just curious.
Sure.
And my original question in terms of bears, do you find that the majority of them, the reason why they're coming into more residential areas is because boundaries, or do you think a lot of it also has to do with the way humans are managing their trash or managing like other things like that too?
It's, I would say trash.
Mainly.
Yeah. There's more trash available all year. That was really only available in August. Now granted, they are in that hyperphagia phase, where they're trying to really bulk up for the hibernation season. But bears are like, hey, there's trash can here that's unsecured. I was looking around, oh, maybe there's an apple tree here.
And then they learn that that's where
Exactly.
there's now there's
Yeah. Bird feeders, chicken houses, bees, that's all food for them.
One thing I can add is that one of the programs that our education and outreach staff have is they actually have bear sitters. So if a bear is located in a tree, either on open space or in adjacent residential areas, folks will sit there and keep the public away, dogs away, until the bear can safely get down. Sometimes we help with CPW with encouraging the bear to move along, but we are trying to proactively keep people away and keep the bear safe. There's volunteer opportunities there.
Kind of to add to Tammy's there, about 80% of the calls that CPW has responded to as over trash cans in particular. And the ordinance, I believe, for the city went into, for the walking trash cans within a certain distance from the mountains went into effect in 2014. Have either of your organizations noticed a significant decrease in town? Because I know that there is a little bit of an eastern migration for bears further in, closer to Denver County instead of Boulder. They're looking for other sources further and further out as time goes on.
There have been fewer instances with that, and I think the trash ordinance has really helped a lot. And the city was, right before COVID, was studying whether to move that requirement further east of Broadway, and COVID kind of slowed that down a little bit, but that needs to be addressed again, brought up again by the city. So Boulder Bear Coalition is hoping to have that happen soon.
We've spotted them on South Boulder Creek, if you guys know the bottling trailhead in that vicinity. And I've heard of reports going as far east as Woodhills. So they're definitely traveling farther far, trying to find those resources.
In regards to restoration, what is the one project that's been most impactful and rewarding to you?
So when we developed our wrestling ecosystem management plan back in, it was approved in 09, maybe 2012, I can't recall. We recognize that our wetland and riparian systems are the most degraded across our system. And so we've got a very large focus on doing riparian restoration in particular. And so my favorite location is actually South Boulder Creek, and you can see the change that have occurred over the last 15 or so years along that area. So we perform obviously woody and herbaceous plant control, this kind of land management, that can be the herbicides or not. We put in trees and shrubs where appropriate. In the case of South Boulder Creek, we've got 2 federally threatened species. We've got the purple-headed jumping mouse and the lady stress orchid. They both have very different habitat requirements. So where we've got connectivity with the uplands, are trying to maintain that as non-shady, so open habitat for the orchid. And in other areas, we are trying to replant native shrubs and also cottonwood trees to provide hibernation opportunities for the house. So that's my favorite place to work. It feels like it used to kind of be our backyard with our offices instead of smooth, but it's just a nice place to be.
Okay, so we got 1 and then 2 and then 3. So
What are the primary causes for beats of restoration that you typically find?
So we obviously have a lot of modification, especially of our riparian corridors. We are trying to restore a more riverine type habitat rather than all the open ponds that you see around town. There are really only like 7 natural open water bodies in the Boulder Valley. The rest have all been the result of gravel mining that occurred as recently as the 90s, with some going back to the 50s and possibly earlier. So we are trying to restore that habitat, so you have that habitat heterogeneity, so a variety of different species can exist across the system. Invasive species are another large contribution. We have a lot of ornamental escapees, things that were planted back in the 40s and 50s, and it took them a few decades to finally get spread around by birds or what have you. And so we do a lot of non-native removal. The other thing that you may notice as you're walking around and you see these restoration law signs, we have a lot of undesignated trails, and most people, one, either don't know what an undesignated trail is, it's one that was not designed, was not intentionally built, it was just possibly a deer path if people started to travel on, or it could be a cow path if people are traveling on. And so we are actively trying to engage folks to stay on the designated trails. That was a challenge over COVID. People were going farther and farther away from one another, trying to stay distant. So those are probably the 3 main disturbances, I would say. And then we'd be slowly, incrementally trying to knock things back into a trajectory such that ideally, as a human restoration ecologist, I can walk away and the system has what it needs to function without continued human influence.
I think Macy was.
So what's your most like interesting bear interaction that you've had just in your career over time working in the?
So when I lived in West Boulder, I had more bears in my backyard than I've ever seen on a regular. So I watched a bear one night go into an ornamental birdhouse and pry it apart. It took a good 30 minutes to get at this teeny tiny house for an egg. So I'm certain that the amount of energy that was expended going after those eggs. And then the poor mom was sitting right by the window, like by our back window. You could just see her like, oh no. But I don't think the counter multiple bears on open space, to be honest with you. But I also don't work that often anymore in the forest. I'm more down in the plains.
Blair, what was your?
So, well, Brenda was telling me this story about how she had a mama bear with 2 cubs. She was bear sitting, and the cubs were just crazy. They did not want to nap. And the mom was like exhausted. She really wanted to nap. And she finally, usually the mom sits below the cubs, and the cubs, when they come down and they wake her up, she swats at them or something to keep them up. But she finally just gave up and she let them come down the tree. And the cubs came down the tree, they crossed the street, and they went into another yard where there was an apple tree. And Brenda watched them for about an hour and a half, rolling around on the ground, eating apples, running up the tree, coming back down. She said that was her favorite.
I will also add that when I do have worked in the mountain parks, it's always humorous to find an apple tree with a whole bunch of bears scattered right beneath it. You can tell the bear is just gorging itself and pooping it right out because oftentimes the apples are not even digested. So that's really humorous to come across.
And then, yeah, can you say your name?
Pam.
Pam.
I live in the foothills, in Left-Hand Canyon, and this year we had just a plethora of rose hips, apples, choke cherries. There was so much food, wildlife, especially there. And so I'm surprised to hear that so many come into town. Are you saying that it's mainly the trash availability?
Well, so this year, there had not been hardly any reports of bears in town. So that was just kind of, that's an overall kind of generalization there. But this year, this fall, we think because there's so much food in the foothills that they're not in town as much.
And I have another question. I assume the mother is training her cubs to go to trash cans.
If the mother has been habituated to trash, yes, she'll do that. Sometimes, a lot of times in the foothills, there's a male bear too, might be more of a threat to the mom and the cubs. And so the mom will come in town because she feels a little safer there. And so that's another reason for the trash getting locked up. It's just because if they find that easy food source, those cubs can learn to go into that.
We have a number of treated bears near our Chapman Road trailhead, if you're familiar with that, going into Canyon. And we have plans to expand the trailhead. And one of the big concerns that our wildlife staff and others on our ecology team have is, should we really be putting picnic tables here? You know, let's make sure that all of the trash cans are lockable. Just trying to be proactive moving forward and trying to consider that rather than trying to provide every amenity that every or any older citizen would want. We're trying to be mindful.
And this is kind of a simple question, but they really recognize refrigerators, don't they? They know a refrigerator. Whenever there's a break-in on campus, they make beeline for the refrigerator.
And the cars, unlocked cars. It's
And
amazing
cars,
how they can get in there. Yeah.
When you said that you saved like 26,000 pounds of fruit, has that been distributed like all across Colorado or?
No, it stayed in, well, actually, I take it back. It went to Louisville, went to the community food share. This year we had a partnership with them, and they came and picked up from our shed once a week. We probably took another load to them, and another time during the week. But yeah, we harvested 26,000 pounds, and so they got most of that. Now we do sort through the fruit kind of and make sure the bruised and kind of bird-eaten fruit goes to goats and bears, the wild animal sanctuary, and pigs. And so all 26,000 pounds is the community food share that they got. And we did have 750 requests for harvest, and we only got to 92. So that's a lot more apples we could have.
Would you kind of push some of the responsibility on like residential people whose land is on? Like, so obviously your team can only be so big, if there's
Right.
trees, and
Right.
if they want to be a part of it, could like, that's kind of a scary question, but would you kind of be able to ask them like, hey, would you pick that, and pick it up or something?
Right, so so they contact us to come up with a demand, and that's a lot of people just have the manpower to do their own tree, you know, and a lot of people they've lived in their house for 25 years, and they're getting kind of older and they need help, and so, we try to encourage like, hey, can you get your neighbors to come over, and maybe we'll drop off picking poles for you guys, but we just don't have, we need a harvest leader or someone to organize it for them. That's something we would, we'd love to try to reach out and get everybody, but it's just a human.
Manpower. So if you are feeling like volunteering, maybe eating some additional fruit next year.
Exactly.
Michelle.
How much lead time do you need if we know clumps of trees that can go to you? Like, if we know when they're producing, we have a lot of fruit trees.
Right.
We live in a mining community up 4 miles. Substantially lost a lot with the flood, but a lot of stuff is coming back.
Right.
And we pull early so that we don't get theirs in the fencing closer to the houses.
Yeah.
What we've been doing is take part for ourselves and then get big bundles and put them near the creek away from houses. They know where the trees are. They have them in our yards and stuff.
Right.
Is that okay to do that just to deter them? Not near dogs, not near, we can't get rid of the trees. And some of them are on the historic thing, so.
Right. There's like a
Gray area.
Yeah, it's a gray area. You're still encouraging them to go there to get the fruit, and they'll, they need to actually. What if it's a bad fruit year? They're just going to be looking at that spot. I mean, they need to kind of, go to their own, find their own places that source of fruit and source of food, I guess I'm saying. This food buffer that we've been planting, that's kind of a natural food source. It's not necessarily supplying it for them because, hey, there's weather and it'll kind of, ebb and flow. So we would just encourage you to, give that food to a farmer or somebody for their animals or whatever, or food share or all this stuff, yeah. So usually, what your first question was, how far in advance do you need? We like people to register their trees like in July or August and let us know whether it looks like it's gonna be a big crop that year. And then, give us another email a couple weeks, say, hey, I think it's gonna be ready in 2 weeks or whatever.
Okay, perfect. Can I tell students one thing? Do you remember when Sam came to talk and we had the sample tree that they grafted and that tree was called Michelle? That's Michelle.
So I think we have time for probably one, maybe two more questions. Yeah. For both of you, how did you end up in your respective lines of work?
So I'm a volunteer. This is just a passion. And it was just that big fruit year. I just saw all those apples and I was like, we've got to be able to do something with these apples. And so I just kind of started Fruit Rescue. And I've been involved with it ever since.
What's your actual background?
I'm a French major. I'm an MBA French major, and I am doing accounting for a small startup.
I actually intended to see you for my undergrad. I have always enjoyed working with plants since my first non-babysit job at age 15. And so, it just kind of was a natural progression. And just the landscape of the Boulder Open Space program is so phenomenal to me that even when the politics gets ugly, you can walk outside and, be with nature.
Any last questions? Let's give our panelists a big. I want to thank all of our community members and the people who contributed to the symposium today. For the students, we're going to do a very, very brief debrief in the other room and take a picture together. And thank you all for attending. Please reach out to us.